Hi, everyone. My name is Denghui Chen. I'm currently a research data analyst from Promet Lab at UCSD. And then today I'm going to talk about a new genotyping pipeline for Peromyscus californicus based on low coverage whole genome sequence. Uh, so when you guys see the title, the first question might pop out in your head is why Peromyscus californicus, aka so it's also called California mice. California mice from the graph here, you can see actually phylogeny is is a model between phylogeny is a really good species intermediate between two major rodent genetic models and humans, and it can serve as it can help us to understand the linkage between mouse and Neuratis by serving as a really good outgroup. And then second, it is an emerging model for a number of psychological and behavior traits. For example, it is widely used for behavior studies, especially in autism. And also it is really good model for type 2 diabetes because they are monogamy, and it's also a really good model for partner fertility studies. Third of all, it has many well-established colony for other animals for experimental purposes, such as the PGSC. And so the only reason why it is not really widely used for genetic analysis is because there are no practical model methods for dense genotyping in this species. So from our lab, we have already developed a really successful genotyping pipeline based on the low coverage WGS for HS rats, SD rats, and then zebrafish. Here, we are trying to take over that pipeline and apply it to California mice. First of all, I want to thank all these people who supplied us all those initial 96 animals. And on the right side is like on the right side is the genotyping pipeline flowchart for our genotyping pipeline. So let's jump right in. There are four main steps. The first one is the sequencing and then and the demultiplex. So for sequencing, we use for sequencing DNA isolation, we do it 96 sample per batch. And then for the library preparation, we use the Riptide library preparation kit, which is based on random priming from both ends. And then it's also 96 animals per library. And then after that, we use the Illumina 916000 to do the sequencing. For the sequencing process, we do 10 libraries per lane. So per lane, we do 960 animals. So it's like really high throughput. And then meanwhile, we also get like a 0.4 mean coverage for the WGS. And the software that we use for the multiplex is called FG Bio, which is very specifically developed by the company who are developing the Riptide library preparation. The second step is the alignment. So alignment, we use the BWA min, which is like a really general step. For the reference genome, we did have some issues about the reference genome because the currently available reference genome from NCBI is only assembled at scaffold level. And then it's constructed with like so many different small pieces. It's not assembled to like a chromosome level. So it was not perfect based on our needs. And huge thanks to all these people from UC Davis who helped us to prepare a new reference genome for Peromyscus californicus. Although currently the new reference genome is still at the scaffold level, but they're still working on it to stitch them back to the chromosome level, and then it will be published to NCBI very soon. And then from here is like a basic comparison between the new reference that UC Davis is preparing and also another species from Peromyscus, which is really close to the one that we are studying right now. On the right side, you can see the current reference genome, the new reference genome that UC Davis is preparing. They're still at a scaffold level, but comparing to the to another species which has the reference genome for the chromosome level, we can see each scaffold actually covers one complete chromosome or multiple in some other cases actually. So which means the scaffold from the new reference that we have right now is actually very good, but they're still working on it to perfect it. And so based on the new reference genome that we have here, I'm showing to you guys two box plots. On the left side is the total of number raised for each animal that we have, and then the right side is like the ratio of mapped rates. You can see the comparing to the old one, the new one has like a big improvement here. That's why we go for the new one. We totally abandon the old one, and then we hope the new one will bring us better results. And then the next step on the genotyping pipeline is the variant calling. So we follow the GATK best practice to do the variant calling. I'm not going to go into the details, but here's the result. We are able to call 28 millions of variants, and then 23.9 million of SNPs. And inside there are 23.2 biallelic SNPs, which is pretty good based on the initial 96 animal that we have. And here's a PCA plot based on the 96 animals. From here, you can see the green dots. Actually, these 8 green dots, they don't cluster together with the with other 88 animals. It was when we look at it, it was because of those 8 green dots are from Glasper lab, and then they were caught in the wild, not bred in the. They were not bred in the from the colony. So we decided to drop those 8 animals because we are still at the process of developing and optimizing our pipeline. We want to reduce the noise as possible. And then after we remove the 8 animals from there, we still get 21.6 million of biallelic SNPs, and the PCA plot looks much better. And then, yeah, so the next step is the imputation step after the variant calling. For the imputation step, we use the software called Stitch, which is sequencing to imputation to constructing haplotypes. It's developed by Robbie. It's developed by Robbie Davis from Cambridge, and then you can visit that paper. This is like a software for imputation without a reference panel. So because we don't have a we also don't have a reference panel for the California mice. That's why we use this one. And so actually before the imputation step, we did some filtering because we want to keep like a set of high confidence biallelic SNPs before we do the imputation. So we also follow the GATK best practice to do the hard filtering for the variants. After, I'm not really gonna go into the detail about these plots, but this is showing the distribution of the filtering criteria that we did on the SNPs. And then after filtering, we still have 19 million biallelic SNPs, which is like a really good number for us to work with. Here are the density plots for 22 relatively longer contexts that we found from the reference genome. You can see they are really, they're sort of uniformly distributed among the chromosomes, which is good. And then a few small issues that you may see, I pointed out here in the small circles. Here, we see like a small gap here on the contact number 6. And then we also see another small gap on contact number 15. Actually, if we go back to the, these 2 small gaps, and also here on that plot, we see there's like a huge peak on the around location 12.5 megabase. And we think the possible reason why we see those issues is because of the new reference new reference genome. They are not at the chromosome level yet. And then there are some cases where multiple chromosomes will stitch together to be one big scaffold. So that might cause the problem of the gaps here. And also there are some inverted chunks inside a contact that is from the new reference, And also there might be some repeated regions inside the contig actually. And then here are the rest of the contigs, the SNP density plot. And on the right side, you can see the number of biallelic SNPs for each long contigs here. Yeah, the number looks good. And then, so yeah, the number looks really good. We get like a lot of SNPs per each contig actually, based on their length. And so so we also did some error estimation for the pipeline. So first of all, I want to say this project is not funded, so we didn't have enough money to do like a deep sequence to construct like a true set for this group of animals. What we did was we chose 16 out of the original 96, actually 88 because we got rid of it. Yeah, we chose 16 of them out of the original 88 to construct like a true set for our data. And then we try to match the distribution based on the colony and then the gender. So here, oh, actually this is 16, sorry. Yeah. So yeah, so the error estimation design is when we do the imputation, we do two rounds. The first round is the original 88. And then the second round, we swap the original 16 that we chose for the control and then use the control to do the imputation again. So that we can compare the concordance rate between these two data sets. So as we saw, we have like so many different contigs. So we chose one with the length relatively long from those contigs. And then we chose contig 3, which has 154 megabase length. And then the number of biallelic SNPs is around 1 million. Concordance rate between the control and the original data set here is 81.4%. Yeah, this number doesn't sound great, but if we remember the reference genome issue that we talked about, if we remember the reference genome issues that we talked about, this is like a good number for us to start with. So from here, I think the possible cause for the low concordance rate is because of the problem of the new reference genome. And then we have like a really small sample size for the imputation software. Because the imputation software here that we're using is like a data-driven, it's a highly data-driven one. And then the imputation, the other thing is the imputation software requires a lot of parameters. And then some of the parameters were based on the number of generation and then the funder information. And then we don't actually have like a very accurate and correct one for this data set. And the other thing is we don't have a reference panel for the imputation. It's a reference panel free imputation software. So with that being said, we think the next step that what we can do is we're gonna wait for the UC Davis group to refine the new reference genome so that we can have like a complete and better reference genome to work with in the future. And the other thing is we're gonna keep optimizing the pipeline to prune the parameters for GADK variant calling that we use, and also stage imputation. Yeah. With that being said, here's the conclusion. So this is probably the very first cost-efficient and high-to genotyping pipeline for Peromyscus Californicus. And then it allows us to open the door to genome-wide association analysis for this species in the future. And yeah, that should be all. Before I close the slides, I want to acknowledge all these people from our lab who provide me a lot of support for this project, and also UC Davis, and then UC Davis people and UC Maryland people who helped us to provide the 96 initial animals, and then the people from UC Davis Genome Center who helped us to prepare the new reference genome. Yeah. Thank you for listening. Any questions?